Okay, let me share my screen. So can you all see my screen?
Yes.
Yes.
Okay. So, good morning, good afternoon. Yeah, Assalamualaikum warahmatullahi wabarakatuh. So, thank you for your all of you for your participation in our World Class Professor Program organized by General Directorate of Higher Education of Ministry of Education, Culture, Research and Technology of Republic of Indonesia. So it is our pleasure to have you all in here in our Zoom meeting here, even we are in online meeting, but I hope that you can enjoy, you can benefit the guest lecture today. So this year, yeah, we have a world-class professor program 2022 with the title, with the theme of empowering of the role of organic chemistry in drug discovery and development. So in this year, yeah, me, Idham from Universitas Gadjah Mada, are collaborating with the Professor Laurent from Aix-Marseille Université in France, yeah, to help to conduct the world-class professor program. So before we start the guest lecture today, please allow me to explain a little bit about the world-class professor program. So the aim of this program is to improve and to strengthen the international network through the world-class professor program between Universitas Gadjah Mada and the reputable university abroad. Yeah, in this case, Aix-Marseille University. The second objective is to improve the research quality, especially in the field of organic chemistry, through the research collaboration with professors from Aix-Marseille University in France, and to increase capacity, insight, and skills of UGM academic community, especially in the field of organic chemistry, and to improve the quality and quantity of the scientific publication from Universitas Gadjah Mada. And the fifth objective is to increase the ranking of the Universitas Gadjah Mada in World University rank. So to do so, there are three kinds of activities in this program. So the first one is the fine-tuning publication. The second one is guest lecture, and the last one is transfer publication. So in today activity, I'm sorry. Okay, so we have three guest lecture. The first guest lecture is today, 8 November 2022. Yeah, we are honored to have two, well, incredible speakers, yeah, who are expert in their field. Yeah, for example, Professor Norio Shibata from Nagoya Institute of Technology, Japan, and also we have Professor Dr. Laurent from France. The second guest lecture will be held 18 November 2022, and we will have Professor Andrea Basso from Università di Genova, Italy, and also Dr. Kamalullah from International Islamic University in Malaysia. And the last guest lecture will be held at 21 November 2022 from Professor Gadjah from University of Barcelona in Spain. Yeah. So for your information, this guest lecture is joined by participants from the students, researchers, and also lecturers from university and also research institute in Indonesia, Japan, Nigeria, Thailand, France, and Spain. And today guest lecture, we have 101 participants. So this guest lecture is conducted in hybrid mode. Yeah. So in online meeting, yeah, is conducted through Zoom platform, while the offline meeting is conducted in Yogyakarta, Indonesia. And before we start, please allow me to read the curriculum vitae of our world-class professor. So Professor Laurent Commeiras, he is a professor of chemistry in Aix-Marseille Université in France. Yeah. So today he is a head of the the head of Department of Chemistry, Aix-Marseille Université. So his research of interest, yeah, is development of new methodology, including formation of polycyclic complex structure and also total synthesis of natural products and molecule of interest. So without further ado, we wish you to enjoy and get the benefit of the guest lecture today. So thank you very much. Terima kasih and merci Bu. Thank you, Mbak Zulfa. Mbak Zulfa. Mbak Zulfa. Okay, so the next event is the opening remark from the head of Department of Chemistry, Universitas Gadjah Mada, Dr. Nurul Hidayat. The floor is yours.
Thank you, Mr. Idham. Suara saya kedengeran ya?
Iya, Pak.
Yes, sir.
Baik. Oke, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, good morning, ladies and gentlemen. My name is Nurul Hidayat Abdilita. First of all, on behalf of the Chemistry Department, Universitas Gadjah Mada, Jakarta, Indonesia, I would like to welcome all of you to this guest lectures series 1 of the World Class Professor Program 2022. I am very thankful to you, Professor Norio Shibata from Nagoya Institute of Technology, Japan, and also our friends, Professor Laurent from Aix, Marseille, France, for taking time out of your busy schedule to attend this very valuable meeting, very valuable guest lecture. And also, I wish to thank to the committee, to Dr. Idham, Darussalam, Mardjan, who have organized this wonderful events. And also, we are delighted to have you with us to participate in this guest lecture. So I hope through this guest lecture event, we all can learn a lot about CF bond from Professor Norio Shibata, Tetrasubstituted carbon from Professor Laurent. Hopefully also we can enlarge our collaborations, our cooperations between Nagoya Institute of Technology, Japan, Aix Marseille, France, and Chemistry Department, Universitas Gadjah Mada. So I will not to take much of your time. So I think by saying Bismillahirrahmanirrahim, I declare that this guest lecture series 1 is officially open. Thank you very much. Once again, welcome and thank you very much for attending this event. I wish all participants good luck, and I think now I will hand it over to Dr. Idham Darussalam to proceed to the next agenda. Thank you.
Thank you, Pak Nurul, for the opening remark. Mbak Zulfa, can you?
I can turn on the. I'm sorry. Is my sound clear enough?
Yes.
My okay. Thank you for the great opening remarks, Dr. Nurul. Now the documentation section. For all participants, please open the camera. Okay, please open the camera. Check.
No, look like because they will join.
Okay, please open the camera and then we will take the photo together. Okay.
Oh, tadi mikrofon dokter on. Iya.
Sudah Mbak Zulfa?
Please the computer can capture the computer can capture the screen. Then you can start from one. You can start from slide 1. 3, 2, 1. Smile. And then go to slide 2. 3, 2, 1. Smile. Okay, you can go to slide 3. 3, 2, 1. Smile. Okay, thank you everyone for this documentation. Now, ladies and gentlemen, we divide into the next agenda. Is the presentation from our special speaker, Prof. Norio Shibata. Okay, now let me present the professor.
Okay, I'm sorry to take the time. So let me introduce our professor today. So the first speakers will be Professor Norio Shibata. Okay, as you can see in my screen in here. So Professor Shibata, yeah, he is full professor of nanopharmaceutical science, of life science and applied chemistry from Nagoya Institute of Technology in Japan. So he obtained the PhD in pharmaceutical science in Osaka University in 1993 and obtained the full professor at Nagoya Institute of Technology in 2008. He has very high index 64 by Scopus, yeah, very interesting. And also he has already obtained many awards. Yeah, I cannot read all of them because very much in here. And then scientific activities, yeah, he has been a visiting professor in several universities, including in France, yeah, University of Rouen, and then the Normandie University, for example. And also he is editorial in board for the Cogent Chemistry and also Frontier Chemistry and Journal Florent of Chemistry, Chemistry Open, and etc. Yeah. And he has more than 353 publications and reviews, yeah, in the international journal, and also more than 16 of books and more than 115 of patents. So without further ado, yeah, please welcome Professor Shibata, yeah, who will present about the CF bond in here. So without further ado, please, Professor Shibata, the floor is yours.
Okay, thank you very much for your kind introductions. So I'm very happy to provide a lecture here. So can I share my slide?
Yes, Professor.
Okay. Okay, can you see my slides?
Yes, sir.
Yes, we can see it.
It's okay?
Yes.
Okay, so I start my talk. So thank you again for the kind introductions. I'm Norio Shibata from the Nagoya Institute of Technology. The title of my talk today is CF bonds, from the formation to the activations. The breakdown of the drug database has becoming the powerful tool to identify the private structure and functional group in medicinal chemistry to find it out the private structure, to find out the organ foreign compound is going to be very important in the medicinal chemistry. Since the first fluoropharmaceutical approach to the market, proinate acetate in 1954, the organ fluorine compound in the pharmaceutical and agrochemicals have been increasingly prominent. So nowadays, more than 350 fluoropharmaceutical and 430 fluoroagrochemicals on the market are estimated to contain fluorine, which account for the 70% of the pharmaceutical and 19% of the agrochemicals on the market contain the fluorine or the fluorofunctional group. So here is a structural example of the fluoropharmaceutical, especially fighting for the infectious disease, such as the dexamethasone for the COVID-19, efavirenz anti-HIV drug. So they are the anti-malarial drug for artemisinin and melflufen. So this is a drug, not the human, but for the dog of the ticks and the ferrets. So if you have the dog in the family, so you might know the brand name of the vector. Okay, so this slide shows the pharmaceutical in the use in the worldwide for the last 3 decades, since 1991. So which categorize them into the 3 groups, such as the new approved drug, small molecular drug, and fluoropharmaceuticals. The number of the fluoropharmaceutical approved in this period is 191, which account for the 22% of the all small molecular drug approved, 839, and 18% of the all new approved drug of the 1,072. So therefore, the around 20% share of the organ foreign compound is pretty high because the organ foreign compound can be rarely found from the nature. So as can be seen on this slide, so every year, 5 to 10 pharmaceuticals are consistently approved. And as you can see here, the recent 10 decades, the number of the pharmaceutical is going to be increased. Okay, so this slide shows a closer look at the data for the last 5 years for the contribution of the pharmaceutical in the FDA US. We've seen here the percentage of the contribution of the pharmaceutical is 34% in 2015 and 23% of the 2016, 25% of the 2017, 50% of the 2018, and 43% of the 2019. So also the 10 pharmaceutical was approved in the 2020. So these data depicted in this graph clearly suggest a promising future for the pharmaceutical in the drug market. Okay, so because of the importance of the pharmaceutical, so many people are focused on the development of the new synthetic technology for the synthesis of the organ foreign compound. But as I mentioned already, more than 350 from pharmaceutical and 430 from agrochemical have been available, readily available, including the very, very complex structure, as shown here, even the stereogenic carbon center. So therefore, we have some new idea to use this fluoropharmaceutical as a precursor for the new drug discovery based on the CF bond activations. I mean, if the selectivity of the CF bond of the complex molecule and can be transferred to the other functional group, we can instantly obtain the new drug candidate. So, however, the selective CF bond activation is very, very tough because of the bond dissociation energy of the CF bond. As can be seen here, the bond dissociation energy of the CF bond, here the sp3 carbon, and 485 kilojoule per mole, while the CH and CC bond are rather less of the 413 and 346 kilojoule per mole respectively. So therefore, if we try to focus to the cleave of the CF bond by the power, for example, the CH and CC bond could be damaged. So therefore, the selective cleavage of the CF bond is really, really challenging. Okay, let's check the case of the aromatic fluoride. So the SciFinder searching revealed that the largest group of the commercially available halogenic RN is the aromatic fluoride. It's quite surprising because we regularly ask our students which is the largest family of organic halide from the commercial source. So regularly we believe the aromatic bromide over the iodide, because the aromatic bromide and iodide are quite familiar with the organic chemists as a precursor for the further transformation, such as the Suzuki-Miyara coupling reactions. However, this is not true. When we check the SciFinder very carefully, the most commercially available halogenic RN is the aromatic fluoride, more than 6 million compounds, followed by the chloride, bromide, and iodide. Why this is surprising data? Because of the stability of the aromatic fluoride. So aromatic fluoride indeed is quite stable, so it's easy to be stocked for the commercialized. So this slide shows the bond difference of the bond dissociation energy of the aromatic halide and their equivalent. As can be seen here, the CI bond dissociation energy of the 272 kilojoule per mole, bromide 336, chloride is 400, and ether on the 419. So CH bond of the benzene of the 472. So on the other hand, you can see here the CF bond of the perfluorobenzene is 477, and the CF bond of the fluorobenzene is the 526. So therefore, it's very, very clear that the CF bond is really, really tough compared to the others. So therefore, in other words, aromatic fluoride is more favorable platform for the long step synthesis. For example, if you require the multi-step synthesis of some target compound, if you use the aromatic iodide bromide as a starting material, during the long step synthesis, these moiety might be damaged. On the other hand, if you selected aromatic fluoride as a starting material, even the multi-step synthesis, it should be survived. Okay, so in other words, cleavage of the CF bond in aromatic fluoride is in generally significantly more and more challenging. Okay, so therefore there are many people start to join in this area for the activation of the CF bond and the further chemical transformation reactions. So this slide is most of the people focus on this area of the, for the transimetal catalyzed cross-coupling reaction of the aromatic fluoride. And most of them, they are focused on the carbon-carbon bond forming reactions under the transimetal catalysis, also using the organometallic reagents such as magnesium, zinc, aluminum, and boron, and so on. And this chemistry is now expanded to the carbon heteroatom bond forming reactions such as nitrogen, oxygen, and sulfur. So one of the most significant achievement in this area is the defluoridation reactions to provide the allyl borate. And allyl borate, you know, it's one of the most powerful and useful stable intermediate for the further chemical transformation, such as the Suzuki-Mello coupling reaction under the palladium catalysis. Okay, so this slide is a similar work of the nickel catalyzed validation of the allyl fluoride via the CF bond cleavage using the in situ generated nickel phosphine complex. And the two groups are independently published in 2015. For example, the Mardjan and the co-workers of the nickel catalyzed validation using the B2 neoprene 2 as a boron sources, nickel COD as a catalyst, cyclohexylhospine as a ligand, sodium phenoxide is a base, THF, 100 centigrade, and 24 hours. So, on the other hand, in the case of the Niwa and Fosoya, they use the B2 pin 2 as a boron sources. Catalyst is the same, ligand also the same, and copper iodide as a mediator, and the cesium fluoride should be the base, and in toluene 80 centigrade for 24 hours. So both papers are independently appeared almost the same time, but surprising the reaction conditions almost identical, which means the reaction is quite sensitive to the reaction conditions. In fact, in their paper, little bit different reaction conditions, always reaction going to be the messy. So moreover, the reaction required high reaction temperature for the long reaction hours. Substrate scope also is rather limited. So therefore, the development of the novel, more efficient methodology for the defluoridative chemical transformation should be joined in this area for the defluoridation reactions. So in this context, we are interested in the some transformation reactions compound as substrate, reagent, and the product. So aromatic silan or the alkyl silan can be nicely converted to something carbon-carbon bond or the carbon heteroatom bond formations, such as the Hiyama coupling or the flaming thermal oxidation reactions. They are the fundamental area of this research. Also, the organosilicon reagents, such as the trialkyl cyanide, trialkyl cellular azide, allylic silan, also the trifluoromethyl silan. We call the blue button percussion reagents. So they're very, very powerful tool to the direct introduction of their functional group into the target molecule. So moreover, the organosilicon compounds have also been intensively researched for the applications, such as the organoelectronics, photonics, and biological active molecule for the drug discovery. So therefore, we can say the organosilicon compounds have greater potential than the others. So there's how we can access to the aromatic silan, for example. So there are several methods for the preparation of the aromatic silan from the aromatic halide and their equivalent. However, the transformation to the aromatic silan from the aromatic fluoride was not exist when we were interested in this transformation. So why this transformation is so difficult? So we can explain based on the bond dissociation energy. If we use the silating reagent for these transformations, okay, what happens? The bond dissociation energy of the CF1 of the sp2 carbon at 526 kilojoule per mole, while the silicon fluorine bond is the 576. It's one of the strongest bond in the organic chemistry. And silicon carbon bond is much weaker with a 318 kilojoule per mole. So therefore, you can see here, the cleavage of the CF bond by the use of the silating reagent is possible, but to produce of the TMS fluoride, not the carbon silicon bond. And it might be generate aromatic plus and so on. So this is the difficulty for the transformation of the aromatic fluoride to the aromatic silane by the use of this acylating reagent. Okay, so we started this chemistry more than 10 years ago, and this is our earlier example of the shear bond cleavage of the asymmetrical synthesis of the organofluorine compound. The starting material you can see here, so symmetrical difluoride, which have the sp3 CF bond here. And the treatment with the bisphenol acetonamide, and in the presence of the synchron alkaloid dichromethane, one of the CF bond is activated by the silicon atom of the BSS, and in the presence of the synchron alkaloid, the carbamate intramolecularly attack at this carbon to provide the cyclic carbamate with the stereogenic carbon center here with high enantioselectivity up to 92% EE. You see the driving force for this transformation can be understandable based on the bond dissociation energy. Shear bond 485 kilojoules per mole of the sp3 carbon, while the silicon fluorine bond is much more stronger. So therefore, the more stable compound can be released. We extend this chemistry for the allylic trifluoromethylation of the allylic fluoride. The starting material, so-called Morita-Bales Hillman allylic fluoride, and the treatment with the Rupert Prakas reagent in the presence of the synchron alkaloid. As can be seen here, the allylic trifluoromethylated compound was obtained with a very high enantioselectivity of up to 97% EE with the release of the TMS fluoride. The key for this transformation of the shear bond activation by the silicon atom of the Rupert Prakas reagent, followed by the SN2 prime type acid for the terminal alkane. And more interestingly, only the one of the enantiomers was nicely converted to the allylic trifluoromethylated compound, while the another opposite enantiomer of the starting material can be recovered. So it's with high enantioselectivity with the kinetic resolutions. So anyway, the shear bond cleavage by the use of the silicon atom is possible, but the formation of the carbon silicon bond should be very, very difficult to suppress of the silicon fluorine bond. So how can we achieve in these transformations? So we found very nice conditions of the trialkyl silane BP as a reagent, nickel catalysis, tertiary butoxide, ligand-free system, room temperature at 2 or 12 hours. So desired aromatic silane can be obtained quite nicely. And more interestingly, this transformation can be expanded to the defluorosylation reactions of the aliphatic fluoride to the aliphatic silane. But you can see here in this transformation, so no nickel catalyst required, only the tertiary butoxide and metal-free system, room temperature 2 hours. So the alkyl fluoride directly converted the alkyl silane in treat. So this is an extremely rare example of the defluorosylation reactions using the acylating reagent. So the detail will be explained for the next slide. Okay, so standard schema shown here. This is the starting material of the aromatic fluoride. The acylated BP is a reagent, 1.5 equivalent. And nickel COD as a catalytic amount, potassium tertiary butoxide of the 2.5 equivalent. Cyclohexene THF mixed solvent system, room temperature 2 to 12 hours. So desired aromatic silane were obtained. The reagent of the trialkyl BP is nicely accessed by the reaction of the trialkyl hydride, iridium catalyst, DTBT ligand, and with the pin2 cyclohexene 80 centigrade for the 12 hours. So anyway, the substrate strobe are shown here. As can be seen here, the wide variety of the pi extended floral length, which have the fluorine here to here to here, are very, very nicely converted to the corresponding pi extended sided compound in good to high yield. As can be seen here, interestingly, the chemoselectivity is excellent. For example, the ether group are survived the reaction conditions, no CH bond activation, even in the presence of the heteroatom, such as heteroaromatics here and here to here. And the sterically demanding position of the fluoride also nicely transformed into the silicon compound. Okay, so more substrate scope are shown in this slide for the defluorescence reaction of the fluoralanes. As can be seen here, the series of the para-substituted fluoralane are very nicely combined to the corresponding compound, almost independent of the functional group here. And more interestingly, as can be seen here, the highly electron donating moiety, such as the methoxy group, phenoxy group, or the dimethyl amino group, accept as a good substrate to provide the compounding good yield. On the other hand, electron withdrawing groups such as the CF3 and CF5, the substrate were not reacted at all. So these results indicate that the reaction mechanism does not include the regular SNAR type reaction because electron-rich compound is more preferable, while the electron-poor compound, no reactions. Okay, so further substrate scope are shown here. Can be seen, silyleads are also survived. Steric hindrance also okay. BP also survived during the reactions. So tertiary amine or the pyridine ring also survived for these transformations. More results are shown here. We next attempt a selective mono-defluorescence reaction using the multiple fluorinated starting material. In this pyridine, the starting material have the two fluorine here. This is a difluorobenzene. This is the tetrafluorobenzene. But if we carefully use the stoichiometry of the reagent, the desired compound are quite nicely obtained in good to high yield. Okay, so we are next focused of the defluorescence reaction of the fluoroalkanes. And we surprisingly found that this transformation does not require the nickel catalysis. Only the potassium tertiary butoxide cyclohexene THL mixed solvent system is enough for this transformation at room temperature, only for 2 hours. As can be seen on this slide, a series of the benzyl fluorides, such as the beta naphthyl fluoride, alpha naphthyl fluoride, also the para-biphenyl benzyl fluoride are very nicely converted to the corresponding silane in good yield of the up to 81% yield. And moreover, the tertiary butyl moiety of the trifluoromethoxy moiety are survived under the reaction condition. We next focus of the same reaction using the secondary benzoic fluoride as a starting material. So methyl cyclohexane normal propyl attached here nicely converted the corresponding secondary cyl compound, alkyl compound in good yield. So here, so benzylic position, but also the pyridine ring here, no problem. We can get the desired silate product from the fluoride in 77% yield. While the other substrate of the alkyl fluoride, which have the rather reactive position of the benzoic fluoride. So we next examine the reaction using the completely unreactive, non-activated aliphatic fluoride. But the result is almost the same, that the desired alkyl silane were obtained in good yield of the 70% yield. And here, in this case, two benzylic position, but in this case, we require the nickel catalysis, but the desired compound obtained 73% yield. So when we attempt the tertiary fluoride here, the desired compound, the silate compound was obtained, but the cyclic group was migrated from here to here, presumably due to the steric hindrance. So anyway, so this process quite applicable for the wide variety of the aliphatic fluoride. Okay, so this advantage of this transformation is quite mild reaction conditions at the room temperature. We therefore expand this methodology for the later stage defluorosylation reaction of the fluoropharmaceuticals. So this is an example of the steroid which have the aromatic fluoride here. Under the same reaction conditions, the desired aromatic silane compound is obtained 41% yield. So the many stereogenic center, also the conjugated cyclic system, okay as a substrate. Also the methyl ether also survive during the reaction conditions. And moreover, this is the pitavastatin derivative having something problematic functional group such as the ether here, ester, also the cyclopropyl or the quinoline here, but very nicely converted the corresponding sided compound in 50% yield. Okay, so let's go to the reaction mechanism. So this slide shows the proposed process for the defluorosylation of the fluorine. So first of all, the reagent trialkyl serine bpin react with the first mode of the potassium tertiary butoxide to provide the intermediate of the A. A was detected by the boron NMR, and also from here the silicon atom of the boron rearranged from the boron to the potassium via the intermediate of the B. Then C is so second mode of the potassium tertiary butoxide approach to the boron center, followed by the approach of the nickel COD of the silicon atom to provide highly nucleophilic potassium silicon nickel complex D with a concomitant release of the potassium G tertiary butoxy BP. So this compound was isolated and detected by the NMR study. Also, this process was confirmed by the DFT calculations. So anyway, so species D should be the real species for the defluorosylation reaction. Okay, let's go to the next stage of the reaction mechanism. So the approach to the aromatic fluoride to provide the nickel pi complex E with the release of the COD as a ligand. And from the nickel pi complex DE, there might be the two possible pathways to reach the compound. Pathway B is quite conventional oxidative addition, transmetallation, and reductive elimination step. However, this conventional oxidative coupling pathway is not ideal because of the two reasons. First of all, the oxidative addition regularly requires the high reaction temperature in order to overcome the Ca bond strong Ca bond energy barriers. However, our chemistry only the room temperature. And moreover, the oxidative addition regularly preferable of the substrate having the electron withdrawing group. However, in this case, as I mentioned, electron withdrawing moiety such as the CF3 and SF5, the substrate completely unreactive. So therefore, we need to think it over the different pathway for the defluorosylation reactions. So therefore, we proposed the intermolecular SNAR type reaction. So from here, the transfer of the pi bond density of the aromatic group to the D bond orbital of the nickel via the intermolecular SNAR type approach of the silicon atom to the here to provide the F. And from here, the potassium cation approach to the nickel to fluorine to activate the CF bond. Then fluorine could be cleaved and over the nickel bond to provide the G, so nickel pi complex G. And the COD from here to here jumped to again on the center of the nickel to release of the nickel COD as a catalytic generation with the release of the potassium fluoride to provide aromatic silane. And potassium fluoride was detected by the fluorine NMR study. Okay, so let's discuss the case of the defluorosylation of the fluoroalkanes. As can be seen here in the fluoroalkanes, the reaction does not require the nickel catalysis. Therefore, the first step should be stopped on this stage, okay? So therefore, the highly nucleophilic species of C should be the reactive species for the transformation. So we believe the second step of the C approach to the alkyl fluoride in the presence of the potassium tertiary butoxide, and the second mode of the potassium butoxide approach with the Laurent center is kind of the 4 members transition state. So it's essential type of essential process to provide the desired alkyl silane with the concomitant release of the potassium G tertiary butoxide BP, which was isolated. So anyway, so we developed the new synthetic methodology for the defluorosylation of aromatic fluoride and alkyl fluoride, as shown here. And this is the first example of the defluorosylation reaction. We published in 2018. After our publishing, several groups joined in this area for the defluorosylation of the fluorine, as shown here. So anyway, so we developed a new method. So we are next interested in the defluorofunctionalization of the alkanes. So defunctionalization of the alkanes, defunctionalized alkane is one of the powerful strategy to provide the multiply substituted compound over the alkane double bond. So therefore we came up with the idea for our reported reaction using the defluorosylation in the presence of the alkane double bond. So we expect that the 3 components into the 1 compound under the similar reaction conditions. Okay, so now I explain the carbosylation of the alkanes using the cyclyl boronate and organic fluoride via the selective CF bond cleavage activation reactions. Okay, so this is the standard scheme of the carbosylation of the alkanes using the cyclyl boronate and organic fluoride via the selective CF bond activations. The reaction conditions almost identical. I already explained in the first part of my talk. I mean, the 2 equivalent of the succinylene derivative, this is a double bond, and the trialkyl BP, 2 equivalent, potassium tertiary butoxide, 4 equivalent, cyclohexane THF mixed solvent system, 2.5 hours, only the room temperature in the presence of the nickel catalysis. So you can see here the substrate scope. So using the styrene, regular styrene, usual styrene as a substrate, so aromatic fluoride also is approached here, while the silicon atom is approaching here to provide the desired compound. So you can see here very high yield of this is 96%, this is the 91, this is the 58 because of the presumably the strict hindrance of the here to here. And this is the 78, 93, 70, and 90. So all the reactions are proceed quite nicely, only the 2 or 3 hours with high yield, almost 90% yield. But during the reactions, our student noticed this chemistry does not really require the nickel catalysis. Okay, so therefore we check the reaction even in the absence of the nickel catalysis. You can see here, even the absence of the nickel catalysis, high yield of the product obtained, while a little bit lower. This is 91% without nickel, 87% without nickel, 42% without nickel, 87% without nickel. This is 90% without nickel, 63% without nickel, also 85% without nickel. All the cases in the absence of the nickel, the reaction proceeded well. And either a little bit lower, maybe only the 5%, it's something almost equivalent, right? But always a little bit lower. And moreover, not only for the ethyl alkyl silicon, but also the dimethyl phenyl silicon, normal propyl tripropyl silicon, also the tertiary butyl dimethyl group also possible to introduce these positions. Okay, so we further examined for this transformation to see the substrate scope. As can be seen here, with or without nickel catalysis, we performed. So with nickel, 91% yield, without nickel 75, with nickel 85, without nickel 73, with nickel 78, without nickel 69. So all the cases, if we use the nickel catalysis, it's a little bit good, maybe the 5 to 10%, but without nickel, it's the same. And you can see here, excellent chemoselectivity profile was observed, such as the ether bond is survived, CF3 survived, the chloride also survived here. Okay, more substrate scope here. So in this case, we did not use the nickel anymore. So the reaction performed in the absence of the nickel catalysis. There's an example of the substrate having a heteroatom here. So all the cases to provide a good yield of the product. So excellent chemoselective provide no activation of the CH bond of the heteroaromatics. Always aromatic fluoride approach this side, while the silicon BP approach this side. Okay, so this is a more example of the carboxylation of alkanes using the styrene derivative. Should be the same. Okay, so we are next interested in this reaction using the styrene derivative, which have the substitution here, in order to provide a product with a quaternary carbon center. Okay, so therefore, this is the result of the reaction which have the substitution here. As can be seen here, in the absence of the catalysis, no nickel here, so high yield of the product were obtained independent of the steric hindrance. So this is the 71%, this is the 90, this is the 51, this is 69, this is 73. All the compounds were very, very nicely accessed in the presence of the substitution here to provide the compound with a quaternary carbon center. Okay, so we next interested in the carboxylation of the alkanes using the cernyl boronate and organic fluoride system via the selective shear bond activation. Okay, so compared to the aromatic fluoride, alkyl fluoride always completely different. But you can see here the result is quite interesting. With or without nickel catalysis, we attempt. In this case, the starting material is a benzylic fluoride derivative. This is a benzylic fluoride, or this is naphthyl fluoride, also the benzylic fluoride in the presence of the electron with angle here. You can see here 89% without nickel, 91% with nickel, 85% without nickel, 83 is without nickel. This is a 91 without nickel, this is 93 with nickel. Okay, so in all the cases, in the aliphatic fluoride, nickel completely the no effect, also the same result. More results are shown here. This is an example of the aliphatic fluoride, non-reactive, non-activated regular aliphatic fluoride here to the here. High yield of the product were obtained with or without nickel catalysis. And more example here, completely non-reactive fluoride here to here to here to provide the desired compound in good yield. And S-methyl moiety here that survived without any problem. And moreover, this is the secondary fluoride. The desired compound were obtained, while the yield a little bit less, so 36 or the 52% yield. Yeah, it's they are acceptable. Okay, so to highlight the synthetic utility of our very mild reaction, defluoro functionalization reactions. So a couple of examples which have the complex structure, such as this is a tocopherol derivative, this is a menthol, this is a steroid derivative, this is also a steroid type, the styrene here. This is the precursor of the liquid crystalline. all the cases very nicely performed under the same reaction condition to provide the desired 3-component defluorosilation product in good to high yield. Okay, so we next interested in the intramolecular cyclization to discuss the reaction mechanism. Okay, so if we attempt the reaction of the aromatic fluoride in the presence of the terminal double bond here, interestingly, the cyclized product with the quaternary carbon center here were obtained at 81% yield and 87% yield in this case, very high. And moreover, another example of the aromatic fluoride having this strain derivative here under the completely the same reaction conditions to provide the desired compound in 95% yield. You can see here, this cyclization provides a product, even the higher yield. So this result highly indicated by the mediation of the radical species. Okay, so in order to discuss of the reaction mechanism, we next examine the reaction in the presence of the radical scavenger. As can be seen here, when we attempt the reaction in the presence of the tempo or equivalent, the yield is dramatically lowered over the 8%. When we attempt the reaction using the 2 equivalent of the tempo, the reaction completely stopped. We also attempt the reaction in the presence of the nickel catalysis, but the result is the same. So 20% of the product obtained in the presence of the nickel catalysis, but in the 2 equivalents of the tempo, completely no reactions. So these results indicate the generation of the radical during the reactions. We further examine the reaction mechanism based on the radical clock using the radical clock experiments. So here the starting material of aromatic fluoride and the cyclopropyl styrene derivative under the same reaction conditions. So interestingly, the silated styrene were obtained in 55% yield, which indicated the generation of the benzylic radical neighboring the cyclopropyl ring. So this result indicates the reaction start of the addition of the silicon, not the cleavage of the shear bond. Maybe the shear bond cleavage is the second stage. We next examine the substrate, which are the aromatic fluoride here and the styrene here. In this case, also interestingly, we go to the cyclopropyl sided compound in 76% yield. So this result indicates the generation of the benzylic radical here, which have the silicon, is first, and the radical cleavage and formation of the cyclopropyl. So based on these results, shear bond cleavage is a later stage, while the first stage should be the addition of the double to the double bond of the silicon radical species. Okay, so let's go to the reaction mechanism. Okay, first step of the generation of the intermediate A is completely the same as I already discussed in my first part of my talk. And from the A, the reaction mechanism should be something different. And the potassium tertiary protoxide approached to this intermediate as a single electron reductant to provide the tri-ethyl radical with the release of the B. Okay, this silicon radical approach to the styrene to provide the benzylic radical intermediate C. And the C going to be joined with the aromatic fluoride or the alkyl fluoride, as well as the residue of this species to provide a transition state here. And in this transition state, CF bond should be activated by the approach of the potassium cation, as well as the interaction between the borate to the fluoride. then the radical species start to the cleavage of the CF bond by a single electron transfer process to provide the desired silicon compound with the release of the tertiary potassium borate. It's isolable. And moreover, we detect some byproducts, such as the dimer of the silicon species could be the isogenerative of the dimerization of the termination of this silicon radical. Also, we got the overreaction. It might be explained by the overreaction of the second mole of the styrene derivative here. So anyway, we developed the very nice reaction of the carboxylations using the cetyl boronate and organic fluoride. So now the first part of my talk of the defluorosylations. Second part of my talk, the carboxylation here. But you might find the second reaction does not require nickel, but first reaction required nickel. Okay, so we should need to check again of the requirement of nickel for the first reactions. Okay, so therefore we re-examined the first reaction published in the absence of the nickel catalysis. As you can see here, with nickel for this transformation, two types of reaction conditions, 86% yield, while without nickel, 59. In this case, with nickel 82, while without nickel 51. In this substrate steric hindrance, you can see here that without nickel 26, while with nickel 74. All the results are same. Summarize, in the absence of the nickel, reaction is going, but the yield regularly much lower than in the presence of the nickel. Maybe the double or the triple decreased. So further example, we attempt for this transformation with or without nickel catalysis. You can see the result without nickel 25% in this substrate, while with nickel 58. This is the result of the without nickel 41, with nickel 65. So result is almost the same. All the cases, if we use the nickel catalysis, there are either regularly the double or even the triple in this case, while the reaction proceeded also going well, pretty well, even in the absence of the nickel catalysis. So therefore, we believe that in the absence of the nickel, the reaction mechanism should include the radical species. While in the presence of the nickel catalysis, nickel pi complex should be the key for these transformations. Okay, so final part, I little bit to discuss of there are one more result for the defluorosylation reaction. Okay, so this is the result we already mentioned, defluorosylation using the trialkyl BP with or without nickel catalysis. Okay, with nickel good to eat, without nickel it is moderate. So in this case, you can see here, this is a cetyl BP. If we attempt this reaction using alkoxy BP, okay, so what happened? So this is cetyl BP, this is alkoxy BP. Yeah. So two questions: the defluoroborylation or the defluoroesterification. So in the literature, the starting material are aromatic chloride and bromide and iodide, already reported by the use of the alkoxy BP to provide the aromatic borate in good yield. This is the literature reactions. Okay, if we attempt this reaction using the fluoride instead of the chloride and bromide and iodide, what happened? Okay, this is really nice reactions. We got the alkoxy compound instead of the borate. Okay, only the difference of the halogen to the fluoride. And this reaction is quite wide applicability. So I can show the detail of this sorry, so formations. Okay, so this is a basic scheme for this transformation. So starting material aromatic fluoride, alkoxy BP as a reagent, potassium HMDS 3 equivalent, DME as a solvent 100 centigrade for 12 hours after optimization of the reaction condition. We obtained alkoxy compound in good heat. So if we use the isopropoxy BP, the desired alkoxy ether in obtaining very good is 96, 86, 83, 77, and 67% yield, independent of the functional group here, even the purity here. Okay, so more example of the esterification of the fluorine with alkoxyboric acid, phenol ester.
Sorry to interrupt. You have 5 more minutes.
Okay.
Thank you.
Okay, so no problem. So we can see here that all the cases, alkoxy group can be quite nicely introduced the desired positions. Moreover, the tertiary butoxy BP also introduced quite nicely to the desired compound. Okay, so more results are shown here. Even in the presence of the complex molecule or the drug-like derivative, very nicely convert to the corresponding alkoxy compound in G. Okay, so this is the end of my talk. So today I introduced the CA bond cleavage based on the serial BP reagents. So this is a picture of our group. So most of the work by Dr. Zhu, he's here also, the doctor. Okay, so one more student, he's not here. But anyway, so thank you very much for the kind attention. Thank you.
Monggo Mbak Zulfa.
Okay, finished. Okay.
Mbak Zulfa.
Okay, thank. Okay, check. Okay, thank you for the very clear and complete presentation, Prof. Shibata. Now we go to the questions and answer section. For all participants, you can text your questions in the comment section. Or you can click the raise hand button and you can ask directly to the professor.
Okay, maybe I can start the Thank you, Professor Shibata, for your very nice presentation. When I have a look on your compound, you are able to synthesize the tetrasubstituted stereocenter. So try to do this reaction in enantioselective fashion. To have an entropure compound, is it possible? For example, to imagine a chiral group of the chlorine atom or something like this. Did you try?
Okay, thank you very much. So you mentioned the Enantioselective reaction, right?
Yeah.
Yes, actually, we attempt a little using the Enantioselective reactions using some chiral ligand, but at the moment, so no successful results. So your question is very good. We should focus for the Enantioselective version of this one. So in the near future. Thank you.
Shibata, since I have several questions for you. So the defluorination reaction for the alkyl fluorine. So if I'm not wrong, you only use the benzyl fluorine. Yeah. Have you ever tried to use another alkyl fluorine, for example, alkyl fluoride beside the benzyl fluoride?
Oh, you mean so we not only for the benzyl fluoride, we use the alkyl fluoride.
Okay.
So you mean, let's see. So here, no, this is the alkyl fluoride.
The first reaction.
Yeah. So this is a benzylic fluoride, but this is alkyl fluoride.
Okay, got it.
Okay, yeah.
Yeah. And then it is interesting to know that you have succeed to deduce the mechanism of the difunctionalization of the alkene. Yeah. So just wondering, I'm sorry. So the selectivity of the difunctionalization is because of the formation of benzyl radical, isn't it?
Also, difunctionalizations, you mean?
Yeah.
Which difunctionalization? I don't clearly understand. Would you please ask me again?
The selectivity of the difunctionalization, as you can see, the aryl fluorine here will go to the benzylic position, and silyl bromide go to the beta position in here. Yeah.
Okay, I see. So you are interested in the reaction which have the both aromatic fluoride and alkyl fluoride.
Yeah.
Okay, so we didn't check the reactions which have the both aromatic fluoride and alkyl fluoride. So, but all the cases, the reaction is very quick. So we are not quite sure, but it seems to be the aliphatic fluoride. It's a little bit faster than that of the aromatic fluoride, but we should check these reactions.
Okay.
Thank you. Thank you.
And the last one for me, yeah, since the mechanism goes through the radical mechanism, I'm just wondering that is it possible to perform the reaction under photocatalysis, for example?
Oh, I see. So yes, our reaction mechanism, yes, I see one of the sales point for this transformation. Our reaction does not require the photocatalysis. Also, the no LED light, it might be some uniqueness. Of course, you are right. Under the photocatalytic system, this type of the reaction could be possible. For example, the Professor Macmillan, they also provide a very nice reaction of the serological transfer reactions. Yeah. But his cases, they use the general in his papers, they use the photocatalytic system to generate the silent radical.
Yeah.
However, our cases, we does not require the photological system. So it's one of the uniqueness for our reaction process. Okay.
Thank you, Sensei.
Thank you.
Mbak Zulfa.
Okay, maybe this is the last question. Thank you very much for the very clear and complete answers, Prof. Shibata. Okay, now we go to the next agenda. Okay, we go to the next agenda, the coffee break section. For the offline participants, you can take the coffee break in the table in front of this room for about 5 minutes, and the online participant, you can still in here. Okay.
Thank you.
Okay, thank you, Professor. Have a nice day. Okay, ladies and gentlemen, our next speaker is Prof. Laurent Commeiras. I will read his CV first. Okay, Prof. Laurent is a full professor in Aix-Marseille University and since 2018. And also he is associate professor since 2004 until 2018. Okay, let's begin the presentation from Professor Commeiras. Now allow me to welcome Professor Commeiras to present the material of Enantioselective construction of Tetrasubstituted carbon silk Stereocenters. Prof. Commeiras, the time is all yours.
Okay, thank you very much. So I'm showing my screen. Did you see my screen? It's okay?
Yes.
Yes, it's clear.
Not yet. It's not good for me. Just okay. Just to try to. Okay, thank you very much. Hello, everybody. So thank you, Idham, and Joseph for this kind of presentation at the beginning. It's always a real pleasure to be with you to discuss about chemistry we are doing in Marseille. Just yesterday, I was looking for some picture in my phone, and I found this picture when I came to visit you for the ICST in 2019, in which I discovered your country, I discovered your university, and also I was very lucky to do this horse riding to visit your city actually. Donc, I spent a very nice moment. And yes, last year we start our world concerning the chemistry we can do around the heterocycle. The first one was a survey of gamma-lactone chemistry, and the second talk was a survey gamma-hydroxylactam chemistry. And for this year, I will continue my survey around the Enantioselective construction of Tetrasubstituted carbon center, starting from a very interesting building blocks, which are hydroxybutyrolactam. So just before beginning, I would like just to sum up my previous work and the two talks I gave the lecture. So this research program began 15 years ago with the synthesis of these 5 heterocycles, which are the gamma-alkylidene butenolide. And we are interested because we can find this moiety in very large natural product. So on the left, you have only one example, but we have several examples in the literature. And this example concerns this natural product, which is the ribolide, which have a very interesting antibacterial. We develop a new methodology, which is based on a palladium-free solvent coupling between this terminal alkyne and this iodoacrylic acid, followed by, and this methodology allow us to obtain a very large library of this heterocycle. So just to explain how this reaction works, we propose this mechanism. So the first steps will be the formation of this carboxylate by treating the carboxylic acid with potassium carbonate. And after, we will have the oxidative addition of the copper iodide into the carbon iodine bond to furnish the copper-3 complexes. After, we will have the coordination of the triple bond onto the copper, and the second equivalent of a base allows us to obtain, to do an exchange of ligand between the iodine and the alkyne. And reductive elimination would furnish this enine, which is actually activated by the copper. And to finish the processes and to obtain our target, we will propose a 5-hexodig heterocyclization to give this vinyl copper, and a proto-degradation reaction would furnish the alkylidene butylenolide. So this methodology is actually very interesting because we can work with a very let's go back. Sorry. We can work with very different primary alkyne. We can work with differently substituted beta halogenated acrylic acid on the position 2 or 3. And what is very interesting is we don't need the use of palladium, which sometimes can be very expensive. So after we are interested in the post-functionalization of this compound, why it's very interesting? Because this kind of moiety possesses an endocyclic bond and an exocyclic double bond, which are conjugated with an ester function. So within this very small heterocycle, we have 3 different functions, and we can imagine to do several post-functionalization. And in our case, we are very interested to regioselectively study the reactivity of this exocyclic double bond by using a cyclodation reaction to obtain spirolactone moiety. So we first start with the well-known and easy handle Jesselder reaction, and we have demonstrated that depending the substitution onto the gamma alkylidene butylenolide, and by using this diene, which can be easily obtained from the ring opening of this benzocyclobutane, we are able to obtain either the naphthoquinone fused furanone compound or our desired spirolactone moiety, this 5,6 spirolactone moiety. This scaffold is very interesting because from this spirolactone moiety, we're able to synthesize the CED-EAF rings of antibacterial natural product, which is lactonamycin. And always from the same scaffold, we were able to synthesize 2 natural products, which are deoxylambetal B and C. So to do 5,6 spirolactone moiety, we also work with the very known 2+2+2 cyclo addition between the exocyclic double bond of this gamma alkylidene butylenolide and this diene, and we find 2 interesting results. If we realize this reaction in an intermolecular fashion, if you realize this 2+2 cycloaddition in intermolecular fashion, characterized by a cobalt complex, so we obtain the desired 5,6 spirolactone compound, in which the cobalt is complexed with the diene because it's a very good ligand. However, if you realize this reaction in an intramolecular fashion, we never observe the formation of this spirolactone moiety. Instead, we observe the formation of the tetracyclic cobalt-3 complexing, arising for the fragmentation of this gamma alkylidene butylenolide. So I don't want to show you the mechanism of the reaction because it's not the goal of this task, but concerning the pathway of the reaction in intramolecular fashion or in intermolecular fashion, we can obtain 2 different products. And at last, we also synthesize 5,7 spirolactone moiety, and for this goal, we we use the 5+2 sequaddition between this 1,3 dipole and the exocyclic double bond. And one more time, we're very pleased to find that this reaction is selective. So after this program, we are interested to work with another heterocycle, which are actually the gamma-alkylidene butyrolactam. And it was described on the literature that if you treat this gamma-alkylidene butyronide by a primary amine, we're able to obtain the corresponding hydroxybutyrolactam, which is the precursor of the gamma-alkylidene butyrolactam, thanks to a dehydrogenation reaction. So the next idea was to synthesize this gamma-alkylidene butyrolactam thanks to multi-component coupling reaction just by adding a third component, which is this primary amine. So by doing the same, by using the same condition as previously, we are very pleased to find that this multi-component coupling reaction can access to this gamma hydroxy butyrolactam in very good yield. As previously, we were able to do a very large library of this interesting scaffold. We can find as well in several natural products by varying the nature of the substituent on the alkyne, the nature of the substituent on the primary amine, and as well on the substituent on the starting iodo-acrylic acid. So with this wonderful body works in hands, we decided to do some post-functionalization to increase the molar complexity of such a compound. And for this, we take the advantage of the reactivity of this hydroxylactam in acidic condition, because in acidic condition, this hydroxylactam can evolve to this corresponding N-acyl iminium. And these N-acyl iminium are a very interesting intermediate because we have a very heterophilic carbon in this position, in this 5 position. And as you can imagine, you can easily add a nucleophile to just to give more geofunctionalized lactam. And as well, if you are working with that nucleophile in your reactor, you can imagine a beta elimination from this CH2 to give the corresponding actylidene butyrolactam. So we are interested to do this sequence, this two-step sequence in a one-pot procedure, including in first the formation of the hydroxybutyrolactam, followed by the post-formalization of this hydroxylactam by an acidic treatment. So we start this research program by the synthesis of this alkylidene butyrolactam. So the idea was to replace the quench at the end of the reaction, which is actually done with ammonium chloride solution, by a quench with an acidic solution. So you use hydrochloric acid solution. So when you quench the reaction at the end by HCl, we are able to promote the formation of this N-acyl iminium. And as I said, with that nucleophile in your flask, in your reaction, we observe the beta emulation to give the corresponding gamma-alkylidene butyrolactam. So on the bottom, we have some selected GR example in which we are able to do variation of the nature of the substituent onto the acrylic acid, onto the primary alkyne, I was the primary amine. So the yield of this product are actually good, because this is a one-post procedure including 4 different steps, and the Gadjah selectivity is moderate. We observe majorly the Z configuration of the in mixture with the 50% of the E configuration. And it is that substituent at the delta G position of the gamma Hidden bifenolide. The yield is actually moderate, but this is the best. Actually, this is the best procedure to obtain such compound in a one-pot procedure. So next, we are interested to add a nucleophile in our reaction, and we decided to do this reaction in an intramolecular fashion by adding. Oh, I lost my cursor. By adding a nucleophile onto the primary amine. Okay, and this strategy is also interesting because, as previously, from a particle point of view, if you quench the reaction with HCl, so we are able to synthesize to have this N-acylaminium intermediate, and by an intramolecular trap of this acylaminium with this nucleophile, we are able to synthesize this fused bicyclic lactam with the construction here of the tetrasubstituted stereocenter. So we started to work with tryptamine, which is a primary amine with an indole moiety. This indole moiety is very interesting for biological activity, and as well because it's very good nucleophile in such amido alkylation reaction. So as you can see, this reaction is very versatile. We can work with alkyl substituent, methoxy substituent, or phenyl. We can also work with different primary alkyne, and all the products are obtained in good yield. Interestingly, this reaction works also with aryl J component in very good yield, and we are also able to obtain this diffused compound, which is actually very sensitive to acid condition in 37 yield. To continue, we decided to put the nucleophile onto the alkene part. Alors, why it is interesting to put the nucleophile onto the alkene part? Because with this strategy, we will be able to obtain the corresponding spirolactam moiety. So, as previously, yes, we quench the reaction with hydrochloric acid. After the formation of the hydroxy, and as previously, this N-acylaminium could be trapped by the nucleophile to give the corresponding spirolactam moiety. So we also work with an indole moiety as a nucleophile, and as you can see, we are able to obtain several spirolactam moiety. In this case, the yields of the reaction are lower than previously because in this reaction, I didn't tell you before, if you work with non-activated primary terminal alkyne, sorry, we can also observe in the first step the formation of this pyranone. And unfortunately, this pyranone is completely unreactive towards primary amine. So in this reaction, we can observe up to 20% of this pyranone. So the yield, or the final yield of the reaction is impacted by this 20% of non-reactive pyranone. So as you can see, we're able to build by an intramolecular reaction this lactam, this spirolactam, this fused tetracarboline lactam motif, in which we are able to synthesize in racemic fashion a tetrasubstituted stereocenter. So our next idea was to develop this methodology thanks to an enantioselective nucleophilic addition to obtain this tetrasubstituted stereocenter in enantiomeric action in enantioselective fashion. So I just will open parenthesis concerning the enantioselective and more generally the stereoselective synthesis. I prefer to use the term of stereoselective synthesis than asymmetric synthesis. We can find in several papers or books, because actually we can find chiral compounds which are symmetric. So the term of asymmetric is not very well for this kind of methodology. So it's better to use the term of stereoselective synthesis. So as you know, the biological systems are actually chiral. So if you want to have a response with this biological system, which are chiral, we often use to have a chiral compound. And the configuration and the stereochemistry of your drugs, of your compound, which interact with your protein or your receptor, can have a different response depending on the stereochemy of your compound. So we have several examples in the nature in which one enantiomer have a typical biological activity, and the other enantiomer are another biological activity. So the synthesis of a chiral molecule and the synthesis of enantiopure molecule are very interesting actually for the biological chemistry. So from a synthesis point of view, so we have different approach to have your compound in enantiomeric pure form. So you can have a stereoselective reaction or an enantioselective reaction. And both of these transformations are based under the substrate control, under the chiral oxalate control, under the chiral reagent control, or under the catalyst control. So what does that mean? If we are under the control by the substrate, what it means that the reaction for the formation of a new stereonucleation center, new substrate, is under the influence of an adjacent stereogenic group already present. For example, if you start with this ketone, we have a stereocenter in alpha of this function. If you want to realize the reduction of this ketone with sodium borohydride, we are able to obtain two diisomers in which the alcohol and the methyl are in a trans position or in a cis position. Okay. If you have an enantiopure compound here, we are able to synthesize one isomer also in enantiomeric pure. So the configuration of this stereogenic center depends on the configuration of this one. And this is the transformation we call under substrate control. The second way you have to synthesize enantiomeric compound is to use a chiral auxiliaries. So in this case, so we start from an aqueous substrate, and this aqueous substrate become chiral by adding a chiral auxiliary into this substrate. And when you attach this chiral auxiliary, the corresponding product, and after we remove the chiral auxiliary to give the chiral product at the end. And in this case, the chiral auxiliary is recovered at the end of the reaction. So the well-known reaction I can give you for this example is the Evans alkylation. Okay, so we use this chiral auxiliary developed by Evans, in which we attach our gear substrate, which is actually achiral. So we obtain this compound, which is chiral. You do the transformation. In this case, this is a stereoselective alkylation, which is directed by this benzene group. As the enolate is plane, the enolate or the electrophile, we approach the enolate on the antiposition of the benzene, so by the top of the face. And after, so you release the chiral auxiliary by using a reduction reaction to give the corresponding alcohol in enantiomeric pure form, and you are able to gyrocycle your chiral gyroxylate. So this strategy is very interesting because we are able to synthesize this compound in enantiomer pure. We are able to recover at the end of the reaction your chiral auxiliary. The main inconvenience of this strategy is you have two steps more from the initial Gadjah synthesis, because you have to attach the chiral auxiliary, and after you have to remove the chiral gyroxylate. Another strategy to have an enantiopure compound, which is based on the chiral auxiliary control, is the use of a chiral reagent. Okay, and this chiral reagent will be able to convert your achiral substrate into a chiral product. Okay, so for example, we can have this reaction. This is an epoxidation reaction in which we use this chiral oxidant to give the corresponding epoxide in very high enantiomeric access. The major innovation of this approach is the use of a stoichiometric amount of chiral reagent, which is actually not very atom economic. And to finish about the stereoselective synthesis, the last approach we can have is an approach which is controlled by a chiral catalyst. This is the same approach as previously. Expected that we don't have a chiral reagent, we have a reagent which is associated with a chiral catalyst. And the beneficial of this reaction is that the catalyst is gyrocycle under your catalytic system, and you don't have to need a stoichiometric amount of this chiral catalyst. For example, I can tell you about the reduction of this ketone by using a CBS reagent. This represent reagent is the catalyst, and the reducing reagent is the H3, which is stoichiometric, and we're able to obtain this alcohol in enantiomeric pure form. So as you can see, actually, this method, which is based by the chiral catalyst, is the most attractive method because it's most economical in use of enantiomerically pure stacking materials. So to just back to our project, so we decided to synthesize this tetrasubstituted stereocenter by using a transformation based on control of the Gadjah catalyst. And to be more precise, we use the concept developed by List in 2006. We have the Contorian dialectic catalysis named acidity, which is based on the use of a chiral ion pair between a cation, which is our N-acyl iminium, and a chiral catalyst, which is actually the anion. So for the catalyst, we have chosen to work with CPA, this chiral diphosphoric acid. Why we choose this catalyst? Because it is a dual jar catalyst, in which here you have a branched acidic seat. Thanks to the acidity of this compound, we are able to form the corresponding N-acyl iminium. And the resulting diphosphonate is a chiral anion, and this chiral anion could be form a pair of ions with this acyl iminium, and thanks to this substituent on this position, which directing the enantioselectivity of this transformation, we'll be able to add this nucleophile onto the electrophilic side and obtain the corresponding lactam in enantiomeric pure. So from the literature, we have two interesting results. The first one comes from the group of Masson in France. And Gerald Masson has demonstrated that it's possible to obtain this tri-substituted stereocenter thanks to the same methodology, starting from this hydroxy lactam, with a very good lead and also with a very good enantioselectivity. And the second example I give you is the work made by Ripping for the synthesis, in this case, of a tetrasubstituted stereocenter. In this reaction, Ripping used a more acidic catalyst, which is this japhoramide. However, even if the transformation work, the yield of the resulting product are not very high. They are moderate, around 50%, and the EE of the enantiopure compound or enantio-enriched compound obtained is not very high as well. We have only, for example, higher than 80% of EE. So it means that the construction of this quaternary tetrasubstituted stereocenter is very challenging. And why it's very challenging? Because this is a very steric inverse center, so it's difficult to synthesize this kind of stereocenter. So we have three main challenges for this reaction. The first one is the stereoselectivity, as I said, starting from this N-acyliminium in order to have the challenging tetrasubstituted stereocenter. In our case, as we are working with hydroxylactam with a CH de moiety in this position, we can observe an intramolecular beta elimination to give, in this case, not previously, but in this case, the undesired gamma-alkylidene butyrolactam. And as well, the last challenge is that in our case, we have an endocyclic double bond, and actually there is no example starting from this kind of moiety, and we don't know the reactivity of this endocyclic double bond. So to be honest, I found only one example of such a compound. This example is from the Jacobsen work, in which he did an intramolecular amidoalkylation starting with this hydroxylactam with an endocyclic double bond. In this case, did not use a CPA catalyst, but he used this two-way jar catalyst, and he obtained this fused polycyclic compound in a good yield, and also in a good yield, 88% of EE is actually good. But what is very important to notice that this enantiomeric excess is very lower than the same obtained with a saturated hydroxylactam. So this is our the four challenges: the stereoselectivity, the chemoselectivity, the unknown reactivity of this alpha-beta saturated lactam, and as you can understand, the intermolecular reaction is always a challenge because it's always more difficult to do compared to an intramolecular reaction. So we start our study, and for this we have designing this model substrate in which we have decided to protect the nitrogen atom with a benzyl group, as it was reported by different groups such as Jacobsen, Whipping, or Wuyang. So we realize this reaction in a classical condition by using dichloroethane as solvent in a concentration of 0.4 molar at room temperature. We first did a screening on different catalysts, starting from the least endangered to the most endangered catalyst with this substituent. And this is the TRIP catalyst. This catalyst we call the TRIP. We obtain the best results in which we have the corresponding tetrasubstituted compound in low yield, which is a 40% actually. Why it's a low yield? Because it's a mixture, one mixture with the undesired echidne butyrolactam. And as you can see, the conversion of the reaction is not total, even after 6 days of the reaction. And at last, the stereoselectivity of this reaction is not very good because we obtain this compound with only 53% of enantiomeric excess. So as you can see, we have a very low stereoselectivity, we have a very low chemoselectivity because we have one mixture of the desired compound and the undesired compound. And as well, the reactivity is not very good because we don't have a full conversion of our substrate. So we decided to do some optimization of the reaction of the condition by varying the nature of the solvent, by varying the concentration of our reaction. Also by working with different quantity of nucleophile, by working with different temperature. We also work with a very more nucleophilic nucleophile, which is actually methoxyandol. We work also with more acidic catalysts, such as the corresponding isophosphoroamide. And to make a very long story very short, because it takes maybe 1 year of work, unfortunately, we never obtained better result as previously, in which this compound will be obtained only with 53% of enantiomeric excess. So from these results, we suppose that the interaction between the catalyst and the N-acetylinium appear not efficient enough. So we wonder if it was better if we add an hydrogen bond between the catalyst and our acetylinium to favorize the interaction between the cation and the anion. And this idea was supported by some recent work did by Zhu and Gadjah, in which they use the non-protected non-nitrogen atom with the endolinum. So we did the same reaction. We did the same reaction with gamma hydroxybutyrolactam without a benzyl protection, a benzyl group onto the nitrogen atom. And starting with this non-protected lactam, and by using the same condition as previously, we are very pleased to find that the conversion of the reaction is total within 8 hours of tearing. We only obtain the compound resulting from the amido alkylation. We never observe the gamma alkylidene butyrolactam. So we obtain this compound in very high yield, 95% of yield. And to finish, the stereoselectivity of this reaction is good because this compound was isolated with 75% of enantiomeric excess. So Milan actually, who are the students who is working on this project, made another optimization of the condition of the reaction, and as well, just to be short, she found that by working with acetonitrile into DCE and with a lower concentration, we are able to obtain the same compound with very high enantiomeric excess of 91% of EE. So at this stage, we are very happy to find the best condition, and to be sure that our proposed transition approach are good, we did some more experimental reaction. So we propose that the catalyst could interact with both nucleophile and with the N-acetylinium, thanks to two hydrogen bonds. So just to check if this approach is correct, we first try with this gamma alkylidene hydroxybutyrolactam with a benzyl protecting group onto the nitrogen atom in the same condition, or in the best condition we have developed previously. And as you can see, if you work with a protected lactam, we only observe 10% of enantiomeric excess. So this hydrogen bond between the phosphonate and the acetylinium will be beneficial. Just to be sure, it's not a problem of a steric advance between the benzyl compound and the phosphonate. We did the same reaction with a smaller group, which is a methyl. And as you can see, we obtain the same enantiomeric excess, the same stereo induction, which means that this hydrogen bond is very beneficial for to have a very good enantioselectivity. And to finish, we also realize this reaction with a protected indole, with the methyl indole. And as you can see, if you work with the methyl indole, the EE of the reaction drop to 64%. And one more time, with these experiments, we are able to prove that the interaction between the nucleophile and the phosphonate is also important. So after we studied the scope and the limit of the reaction by varying the nature on this part, in red part, from the terminal alkene, this part in blue from the starting beta acrylic iodo acid, and also from this indole. So I just show you some selected examples. So this reaction tolerates several functional alkene. We can work with alkyl alkane. We can also work with aromatic alkyne, with a benzene who have a very high EE of 97%, and we can also work with a fan with a lower enantiomeric excess. We can also work with a protected alcohol, methoxybenzyl or OTBS group, also with very good enantiomeric excess. And interestingly, we are also able to work with this very sensitive ketal function in which we obtain the corresponding product in high yield and also with a very good enantiomeric access. We can also work with different substituents onto the endocyclic double bond. For example, we can replace the methyl by the butyl or phenyl group, and we obtain as well a very good enantiomeric access. We can work with that substituent on the endocyclic double bond. And very interestingly, we can also work with an alpha halogenated hydroxybutyrylactam as a starting material, in which this is very interesting because after we can do some alpha phosphorylation thanks to cross-coupling reaction. And in this case, we obtain the best enantiomeric excess of our methodology, which is 99% ED. After we work with different indole, with different substituents on the indole, this reaction works well with halogenated compound, halogenated atom, on position 5 and 6 to give the corresponding product with high EE. We can also work with protecting alcohol on this position 7. And the limit of the reaction is obtained with electron withdrawing group, which can be a neutral function of a cyano function, in which we never observe the formation of this tetrasubstituted compound, but we only observe the undesired product, which is the gamma-alkylidene butyrolactam. And to finish, we also work with an indole which is substituted at the position 2, in which we obtain the desired compound in good yield and also in good enantioselectivity. So in this slide, we can see that we have attributed the stereochemistry of this tetrasubstituted stereocenter. This stereochemistry was established thanks to an X-ray diffraction. However, we can use a model developed in 216 by Goodman, based on nucleophilic addition of nucleophile onto the immune compound approach called type 1 or type 2, in which the different substituents are placed and depending the size of the substituent, they are placed onto the more steric bulky and the less steric bulky compound. In our case, with this kind of acyl iminium, we are in type 2 because the red substituent is a small substituent, the blue substituent is a large substituent. We are working with a non-symmetrical nucleophile, which is the indole. So starting from this model, starting with this model, the more bulky substituent we replace further the bulky Swiss substituent on the J-catalyzed, and the indole, we approach the electrophilic center on the C face of the N-alkyl iminium to give this compound with the right stereochemistry. So the stereochemistry of the reaction could be as well predicted by the Goodman model, and this was confirmed by an X-ray. Two more slides before finish my talk today. So we also do some scale up of the reaction. So all the example I shown you I've made on 0.15 millimole of scale by using 10% of catalyze. We can do the reaction with on one millimole scale, so practically 8 more tenths. And by using the same quantity of the catalyst, we have exactly the same G selectivity. We have the same enantiomeric excess. the same reactivity and as well the same yield of the reaction. We are also interested to work with a lower quantity of catalyst by using only 1% of catalyst in 1 minimal scale. And one more time, in this condition, we have the same selectivity. However, the reaction time is longer, 2 days. So in this case, we observe up to 10% of the undesired gamma-liquidine butyrolactam. This is why the yield of the reaction is lower than previously. However, the stereo induction is exactly the same. And to finish, we also did some post-functionation of our tetrasubstituted butyrolactam. The first one we did is a reduction of this endocyclic double bond to obtain this saturated lactam in only one stereoisomer in pure form. And as I said previously, starting from this alpha halogectin butyrolactam, we are able to synthesize a polyfunctionalized lactam in position 3, 4, and 5, thanks to the key cross-coupling reaction. And actually, we are working on the reduction of this double bond using the same condition as previously to obtain this saturated lactam, which have 3 contiguous stereogenic stereocenter. Okay, so this is all for today. So I would like to thank Milan Seda. I think she's connected with us today. We have done all the work on the enantioselective transformation. So thank you, Milan, for this very hard work. As I said, she spent a lot of time for the optimization of the reaction. It wasn't a very easy task. And also, I'd like to thank the group in which I am working, the Aix-Marseille, and you for your kind attention.
Okay, thank you very much, Prof. Commeiras, for giving such an informative and interesting presentation. Now we come to the questions and answer section. For anyone participants who have a question, you can text in the comment section or click the raise hand button so you can ask directly to the professor.
Excuse me, can I ask a question?
Yes.
Okay, thank you. Bonjour, Professor Commeiras. My name is Hamzah Diksaifrofi, and I am from Universitas Gadjah Mada, Indonesia. And my question is, how did you choose the acid catalyst for the intramolecular or intermolecular cyclization reaction? Can we use other acid catalysts or maybe Lewis acid catalysts such as boron trifluoride diethyl complex to form the N-acyl-iminium ion intermediate? Merci beaucoup, monsieur.
So we can use this transformation with Lewis acid. It was already done by several groups by associating a Lewis acid and as well a chiral Gadjah catalyst. We have chosen CPA because, as I said, we have a catalyst which has a dual Gadjah function. It's at the same time an acid catalyst, so we can do the formation of iminium, and as well as the chiral catalyst, so we can do some enantioselective transformation by using the concept of ICDC.
Okay. Okay. Anyone want to ask any questions?
Maybe from me, Mbak Zulfa.
Okay, please.
Professor Commeiras, have you ever tried to perform the enantioselective intramolecular amido alkylation?
No, not yet. So we started the program 2 years ago, 3 years ago. So we started with the more challenging transformation, which is an intermolecular reaction. So the next step will be to work with different nucleophiles. So this is a program which is in progress, and after we will realize this reaction in intramolecular fashion. We do 2 or 3 examples with amino alcohol. So this reaction is actually works actually, but we need some to optimize the reaction condition.
Okay, thank you. Maybe anyone want to ask some question, or it's clear enough, everybody? Okay, maybe that is the last question from Mr. Idham. Thank you very much for the very clear information and presentation that you share with us today. Maybe we can meet again in next time, and have a nice day, Prof. Commeiras.
Thank you.
Okay, thank you. Okay, well, after 2 presentations from 2 remarkable professors, now I would like to invite Dr. Idham to give us some short closing remarks. Dr. Idham, the time is all yours.
Thank you, Mbak Sulfa. Yeah. So before I close the meeting today, so I would like to thank you again for your participation during the first guest lecture of World Class Professor Program. So, and also let me share. Okay. Also, I'd like to say thank you for our 2 remarkable speakers. Yeah, Professor Norio Shibata from Nagoya Institute of Technology, Japan, and also Professor Commeiras from Marseille Université in France. Yeah, for the interesting presentation. Yeah, and then next, I would like to invite you to the second guest lecture, which will be conducted at 18 November 2022. Yeah, again in the hybrid mode. Yeah, in the online mode by using Zoom, and also by through the offline mode, we will conduct it in the Himay Hotel at Yogyakarta, Indonesia. So we will have 2 speakers. The first one is Professor Andrea Basso from Università degli Studi di Genova, Italy, and also Dr. Kamarullah from International Islamic University of Malaysia. Okay, so we are looking forward to meeting you in our guest lecture. Thank you very much. Saya kembalikan ke Mbak Sulfa.
Okay, thank you for the concise closing remarks, Dr. Idham. So see you all later, and thank you very much. Wassalamualaikum warahmatullahi wabarakatuh, and see you next time to the next WCP.
Thank you very much.
Thank you.
Thank you.
You may.